All right, folks, we've had the setup. It's time now for protein synthesis. Now, you'll notice I'm very deliberately separating protein synthesis and its two processes from replication. Remember, replication is only for the purpose of cell division, and protein synthesis is part of the day-to-day activity of DNA and RNA within the cell. I've posted here a couple pictures to help you conceptualize what we're talking about in this process. You may remember from Baby Bio that this is 2 steps, right? And the steps are transcription and translation, okay? So notice in transcription, the information is passing from DNA to RNA. And specifically, when we think of this, in reality, all RNAs are made from DNA through transcription. But for the purposes of protein synthesis, let's think of this as messenger RNA that we're creating. So we're trying to copy a gene for how you make a protein off of the DNA molecule. So that's what transcription is. This is DNA to mRNA. And then the process of translation is where you take that mRNA message and you convert it to proteins. And this is what we call the central dogma. I'll write that down here. And this is about how information in cells flows. Information in cells flows from DNA to RNA to proteins, and then that's going to give us traits for the organism or phenotype of the organism. All right, so that's what we're looking at here. Transcription, this process takes our DNA molecule and converts it to RNA. And then translation is going to take that message from the RNA and convert it to proteins, specifically a sequence of amino acids, and you know from our biochem unit that that means proteins. So the information from DNA sequences through this central dogma converts to phenotype. And when we say phenotype, what we really mean are traits, right? What you can see when you look at the organism. All right. Functionally, then, let's look at what that looks like. Down here, we're looking at how the genetic code actually predicts an amino acid sequence, right? So we've got these DNA sequences, and remember, DNA sequences are complementary. DNA has both a sense strand and an antisense strand, right? So the strand that contains the gene might be this strand right here, this top strand. That's the sense strand. But what we want our mRNA to contain are codons, right? Basically the mRNA equivalent of the sense strand. And we do that through complementary base pairing the mRNA nucleotides to the DNA nucleotide. So we don't copy mRNA from the sense side because we want the mRNA to basically read almost identically to the sense side. It's just identical except that the T's are now used. So what we do is we copy it from the antisense side of the DNA molecule. Okay, and that way the mRNA molecule that we create has the codons, right? So the codons are essentially identical to the sense side. And if you think about it, if you were to cover this up right here and look at it, just like cross it out, notice that the codons on the mRNA molecule are identical to this top strand on the DNA, right? So you're binding it to the opposite side because you're creating it through complementary base pairing. All right, so mRNA contains the codons. Now you're going to see in translation that we need to bind these codons to anticodons on tRNA, and again, those are the opposite of the codons. You'll also remember, oh, and by the way, this is just, all this that we're looking at is complementary base pairing. So if I erase this, you can see that these codons that you have are complementary. This AUG that you see here is complementary to the TAC, right? Okay. All right, so essentially, if you can take a DNA molecule, complementary base pair mRNA nucleotides in, then you can actually read using a chart like this every 3 letters, which is a codon, and you can get the sequence of amino acids that's going to make the protein. We did look at this in baby bio, but to do this, really what you do is you always read the first base here. So, like just reading our sequence here, AUG. Here's the first base in the codon, A. The second base is read across the top, U. So where the A and the U come together is in this box. The third base is read from this side, G. AUG is methionine, MET. It's also a start codon. Okay. In other words, that's kind of telling the ribosome attached to this side of the RNA, the mRNA molecule, and start reading here. All right, so AUG. Let's do another one just to give you some reference here. Okay, GCC is the next one. Here's the G, here's the C, and here's the third C right there. So the G and this C come together in this box. The third base, this C is the second line. GCC encodes the amino acid alanine. Okay, and that's what you're seeing here. So met, ala, and you can keep going. Hopefully that's enough, and if it's not, just ping me during office hours. I'll answer questions for you about that. But that's how you would go about reading one of these charts. All right, now let's look at what actually happens during transcription. Now remember back in the DNA and replication and RNA video, I told you there were 5 enzymes that you had to know. One of those enzymes came from transcription, and that enzyme was RNA polymerase. So the enzyme we are looking at here, and just like in replication, there are multiple RNA polymerases, but all you need to know is RNA polymerase. And this is just like the DNA polymerase, this is the enzyme that's going to add the nucleotides to the growing strand. It's just that in this case, RNA polymerase is adding RNA nucleotides. DNA polymerase would add the DNA nucleotides. Sometimes this is abbreviated as RNA pol, just so you know if you ever see that in a question. All right, so transcription is really easy. If you understand translation, you understand transcription, right? So this is the first step of protein synthesis, and you can think of this as getting the instructions from DNA, okay? And again, I mentioned this really makes any of the RNA molecules, we typically think of this as making mRNA, but tRNAs that hang around inside of the cell and RNAs that are part of the ribosomes are also made by this process. It begins at a section of the DNA of the gene called a promoter, and it ends at a section called the terminator. So these are non-coding regions of DNA, right, that allow for the attachment of enzymes that are going to do basically what you saw happening in replication. Notice you've got a replication bubble here, right, and you're going to make a copy of the mRNA, but notice again, this template strand, remember that template strand is the antisense strand. So this is the antisense, or it's usually abbreviated AS. And the non-template or coding strand is the sense strand, meaning that it's coding. This contains the actual copy of the gene, but notice we're binding the RNA to the DNA to create it. And if we want the RNA to read the same way that this sense strand does, we've got to bind it to the antisense strand, right? And the mRNA is made in this direction from 5' to 3' when it's made. So it looks just like replication. You're using enzymes to open up the DNA molecule and so on, except that you're only going to copy one gene, so you're not going to have multiple transcription bubbles happening that would ultimately unzip the DNA completely. So that's not going to happen here. You're just unzipping one gene and copying the one gene. And then after that, you're only copying one side. You just need the side that you can complementary base pair to. And so that's, if you look at this arrow, that's what this is showing down here. You're making it in this direction. Okay. And that's about it. At this point, what you should probably do is go ahead and pause. There's another video that I have linked in Google Classroom for you to watch so that you can see the animation of this. So go ahead and pause the video here and watch that animation, because it does help. These are moving processes, so it does help to see these in real time. All right, so you're back. So what you saw in the video is a bunch of transcription factors that kind of assembled at this promoter region that you're seeing here on the DNA molecule. And when we get into the DNA regulation video that I'll make next, we'll talk about those transcription factors. But essentially, you get this RNA polymerase. There it is. That's the one enzyme you have to know from this. And RNA polymerase is going to attach, and notice the template strand is right here. This is the strand that's going to get copied, and it's being fed right into the enzyme right here. Here's our intake valve, and it's putting RNA in, and as the RNA is created, it snakes out of the top. And the RNA polymerase just kind of moves along, unzipping and building RNA. When it reaches the termination sequence, there's a termination signal encoded on the DNA at the end of the gene. Then the RNA folds into this hairpin turn, and it gets released and ejected out. And you saw all of that in the little video. All right, now that is not all, though, and this is where it gets a little different from what you saw in baby bio. Okay, that's the basic process. But there are some things that end up happening when we finish making RNA, right? So after transcription, this new molecule that we've made is actually called pre-RNA because it's not ready for translation. First of all, you have end modifications. So the 5' end gets what we call a cap. It's guanine rich, and it's about 20 to 40 nucleotides that gets attached to this end. And then at the other end, you get the poly A tail. And again, this is a bunch of adenines that are added to the end. It can be anywhere from 30 to 250, and essentially this aids in getting this, when it's finished, this molecule out of the nucleus so that it can head to the ribosome to be translated. So there's that as well. So oftentimes genes have these coding regions and untranslated regions. So the coding region is the actual code for the protein that we need. But stuck inside, and sometimes even stuck right in the middle of a coding region, you could have a section that is not needed in order to make a protein molecule that we need. And we need to remove those non-coding sections. And some of you are saying, why in the world would they exist? Scientists still aren't exactly sure. There are a couple of hypotheses for this. One hypothesis is that these non-coding regions actually extend the length of the gene, which would increase the probability that crossing over back in meiosis would occur here, so that you could get more genetic variation in that gene. Some scientists think actually that the RNA can be differentially spliced to make, so you have one gene, but depending on how you splice it or remove the non-coding regions, you can actually make different proteins out of the same gene. So it's for efficiency, so that the code is more efficiently written that way. And again, we'll get into that into the next video a little bit later on. But what you need to understand, there are two terms here, exons, right? Exons are what we call the coding region. So this is an exon, and it's kind of counterintuitive because you think of an exon as, that EX makes us want to remove it. So I always think of this as the exon exits the nucleus, okay? So these coding regions are also called exons. Introns are the in-betweens. Okay, these are the regions that are not coded and have to be removed. And this is a really detailed diagram about how that works. I mention this here because I like to say SNRP. What actually does the removal of the introns is called a small nuclear ribonucleoprotein, which joins to some other factors to create this spliceosome. But this SNRP, or small nuclear ribonucleoprotein, It is just a fun word to say. So there it is. There's the SNRP, possibly the most fun word in biology. But it creates a spliceosome, and this just shows you essentially what the spliceosome does is grabs hold of the ends of the intron, slices here, slices here. When it slices this side, it attaches it to the cut end to the other side of the intron and then removes it and then joins these two pieces of the exons back together. Okay, that's basically how it works. You don't need to know particulars here. But essentially, so here's our promoter, right? Here's our terminator sequence here. And if you have this, if this is the gene that we're looking at, there's an exon, there's an exon, there's an exon. This intron has to be removed and this intron has to be removed. So the primary transcript looks like this. Oh, sorry, and this is the DNA. So the primary transcript from that would look like this, right? Exon 1, intron 1, exon 2, intron 2, and exon 3. So these little light sections would be removed by spliceosomes, and then the splice transcript is here. So essentially, there are a lot of times they'll ask questions about what's the relationship or compare the primary transcript from transcription to what actually is translated at the ribosome. One of the things is you have a cap and you have a tail, right? The other thing is that the gene itself, the primary transcript is much longer than the spliced transcript. All right, and now I'm going to explain this, but again, after this, I'm going to want you to go ahead and pause and watch the video segment of this. But first, let's just look at what's going on. First of all, this little tangled mess is what I call the spaghetti model of a ribosome, but this is just, you know, it's just a chemical model of a ribosome. Here it is, not as a spaghetti model, but with the ribosome as a solid organelle within the cell. Notice that this ribosome actually has 3 chambers. In baby bio, we focused you on 2, right? You have the E, this is where this is the chamber where the tRNA molecule is going to exit, and these little things here, these are the tRNA molecules. In the last video, I showed you the cloverleaf model, or the 2D projection of a tRNA, but those tRNAs actually twist and fold even further to make them actually have this kind of shape. But essentially, you've got that. This is the primary site. This is the A site, the aminoacyl site. But the P site, this primary site, is where the polypeptide is going to always remain attached. So anyway, so you actually have 3 chambers here. Notice the mRNA, right, is fed into the ribosome. And the ribosome has 2 main parts. This is the large subunit. This is the small subunit. And it's the large subunit that has the chambers for the tRNA. Notice also when a codon sequence is underneath the P site and a codon sequence is underneath the A site, the anticodons, the tRNAs that have the anticodons that match those sequences drop in. And the key to this whole thing is that each anticodon sequence that you see here calls for a specific amino acid to be attached at the other end of the tRNA molecule. And I mentioned this in the last video, that every tRNA is folded up so that one anticodon shows, and then the other end of the molecule is where the amino acid can attach. Okay, and the key here is that this anticodon matches a specific amino acid, and it's that matching that makes sure that the correct amino acid is being brought in at the correct time, because the tRNA can only come into the ribosome when the anticodon it has matches the codon sequence on the mRNA molecule. So here's the P site, here's the serine attached, another one comes in, this is the A site, and this particular amino acid is alanine, okay? Now, once this situation occurs in the ribosome, right, everything shifts. Specifically, really what happens is the ribosome moves in this direction along the RNA molecule, the mRNA molecule. That's called translocation. Okay, so as the mRNA gets pulled through the ribosome, or the ribosome moves along the mRNA, whichever way you want to think of it, that's translocation. All right, so what happens essentially is this. Let's start here. So we've got this empty ribosome, we've got the mRNA attached. The first tRNA comes in, it's got methionine, which that's the start codon, so pretty much all the proteins are going to start with methionine. The second tRNA comes in, there's it's glutamine or glutamic acid, I can't remember. I think it's glutamine, or maybe it's glutamate. Anyway, I can't remember what that abbreviation means, but it's another amino acid. Now we've got a situation where both of these are in position. The key here is that a bond can form between these two amino acids now. Once that bond forms, the ribosome translocates, and it's going to move this tRNA that's in the P site over to the E site, and that's what you're seeing here. And then the A site, the tRNA that was in the A site shifts over. Now notice, whatever's in the A site will now take over holding this growing protein chain as it moves to the P site. So still this tRNA is still holding on to its glutamate, and it's keeping this protein in position here. All right, now you've got an empty chamber. We're moving back over here now. Okay, so we've gone from here to here. A new tRNA can drop in whose anticodon matches this codon. Another bond is going to form. It's going to shift everything over again, right? So that's going to kick out what was in the E site, move the thing from the P site to the E site, move the thing from the A site over to the P site, and again, this is still holding on. And so you get this shifting over, everything shifts over, one thing comes out, and then a new tRNA drops in here. So you get this shift out in all the way down the line until you reach a stop codon. When you reach a stop codon, then that's the code for the ribosome to go ahead and let go of everything that the protein is finished. All right, I just want to make one quick word about prokaryotes in this process. They do these processes. They do transcription, they do translation. But prokaryotes do not do post-transcriptional modification. As the RNA is coming off of the chromosome in, or the chromatin in a bacteria, there's no nuclear boundary for the mRNA to leave. So instead, as that messenger RNA is coming off of the DNA molecule, a ribosome can clamp down and just start working. Okay, so transcription and translation are what we call coupled processes. A lot of times the question will say that they're coupled. And what they mean is that these things can happen simultaneously. All right, the last word on this topic is mutation. Okay, and you're going to do an activity where you're going to explore all about mutation as part of this today. But I just wanted to kind of mention a couple of things. Anytime you have a change in the sequence on a DNA or an RNA molecule, we call that a mutation, right? So that's what that is. A code change is a DNA mutation. Now, that's not to be confused with chromosome level mutations. I put these down here to show you that just for comparison. Chromosome mutations happen like when you looked at a karyotype and you saw that there was an extra or missing chromosome, or a piece of a chromosome was missing or something like that. There are also a couple others, like an inversion where a piece of a chromosome flips over, or a piece of a chromosome breaks off and flips itself over onto another chromosome. Those are all chromosome level mutations. Remember, that's condensed chromatin. These are huge, sometimes catastrophic mutations, because you're messing with big, huge segments of DNA in a chromosome, right? Remember, DNA gets condensed. It gets coiled and coiled again, and coils get coiled to make supercoils, right? So if you take a piece of a chromosome, you're taking an awful lot of DNA, right? What we're going to talk about now and focus our attention on, and let me just cross that out then. That's not, we're talking about just DNA mutations here, not the chromosome kind, where you can have situations where there's a mistake in the sequence itself. So point mutations are changes that occur at one point. The first kind is called a substitution, right? The second kind is the frame shift mutation, which can either involve an insertion or a deletion. Okay, so a substitution is where one base is switched out for another base. So if you look at sickle cell disease, right? So here's the hemoglobin. I think it's the beta segment of hemoglobin. I can't remember, but one of the hemoglobin amino acid chains, there are actually 4 there, but one of the amino acid chains has, the original should say GAG, the mutated version says GTG, right? So this is the mutant version. That's a substitution. And that substitution mutation can take normal plump red blood cells and make them sickle like this, okay, turn into a sickle shape. You can imagine, we normally think of that as not being all that bad, but in reality, when you get a frame shift mutation, it's way worse. And in terms of what it does to the sequence. In reality, if you look at this, I don't think you could tell somebody with sickle cell disease that a point mutation, a point substitution, isn't that bad, right? That makes a big change to the blood cells, and then it causes a bevy of other symptoms that can put someone with sickle cell in and out of the hospital for the rest of their lives. So be careful about thinking, well, that's not that bad, you just change one base. In fact, sometimes these little substitutions are not at all bad. They're what we call a silent mutation, where if you go back to the codon chart, you'll notice there's a lot of overlap in the codon chart. In other words, you might have 3 or 4 different codons that code for the same amino acid. If you make a change, so if we take this TAT and change it to TAC, if you look back at your codon chart, that's still going to encode the amino acid tyrosine. So the sequence is mutated, it's changed, but that mutation does not actually change the amino acid sequence, so you don't get any of these effects. That's why this is referred to as a silent mutation. Okay. Then there are a couple others. These get kind of, the missense and the nonsense mutation get a little specific. I don't know that College Board is going to ask you about that, but what I do want you to see is what happens when you have a frame shift. In this case, if we take our TGG, our original sequence, and we insert a C between the T and the first G, notice that it shifts all the other letters down. So that you start out okay, here's tyrosine, but now we've pushed all the other letters down one space, and what's happening is every single amino acid from that point forward ends up being changed. So these frame shift mutations are particularly nasty because from the point of the mutation onward, that's it. You get a protein that is not in any way resemblant of the original protein we were trying to make. The thing about the nonsense mutation you see here, and like I said, you should know this in general without necessarily having to worry about it being called a nonsense or a missense mutation. If you have a mutation, either a frame shift or a point mutation that results in an early stop, that obviously would be pretty nasty as well, right? Your protein is going to be really, really short. So any kind of mutation that affects the stop codon, either puts one in early or maybe doesn't encode a stop codon, that could even lock the mRNA into the ribosome until it degrades. So all of those would be pretty nasty, and that's what mutations do, although not all of them do. Remember, you've got the silent mutation. And some of these mutations, remember, we normally think of this as bad, but remember back to when we talked about evolution, mutation also fuels some variations, and sometimes those mutations can be good then because it causes a variation that maybe helps an individual to survive. So that's mutation, and that is protein synthesis.